Hi, everyone. Welcome back to the ZEISS Micro Minute. I'm Nate, here again, and I'm excited to talk to you today about ZEISS at the Neuroscience Conference and the cutting edge of neuroscience, the ZEISS Microtome in the Volutome. So first, I want to mention the Society for Neuroscience Conference is coming up this Saturday. That's October 5th, running through the 9th. We'll be at booth 653. This is in Chicago. Now, you probably won't all be there. However, if you are, please check us out. I have a bunch of good friends who will be there, and I will be doing demos, but here from our demo center here in Dublin, California. So at the ZEISS Microscopy Customer Center, and that's where I'm coming to you live today. What we're here to talk about is the cutting edge of neuroscience. Now, this is a bit of a pun that hopefully you'll understand here in a second. And this technique that I want to teach you about, which is become huge for neuroscientists today, is serial block face imaging in the scanning electron microscope. So what does that mean? Let me explain. Typically, if you think of doing imaging of biological samples in a scanning electron microscope, you might think of images that look like this, where you're seeing maybe the cellular surface of the outside. You can zoom in and see details, but unlike a light microscope, you can't see a living cell, you can't see what's going on inside unless you cut the sample. Now, if you cut the sample with certain preparation techniques, with staining, you'll get a nice two-dimensional view of what's going on inside the cell. And now this technique is very important for doing disease, where you're looking at it for what's the word I'm looking for? You're looking for doing diagnoses of disease. So this would be with transmission electron microscopes, also scanning electron microscopes. But what happens if you want to see the full volume? Especially when we think about neuroscience and you're trying to understand the brain as a whole, just seeing two-dimensional images isn't going to get you the data you need. Now, how can you get three-dimensional data out of a scanning electron microscope? That only takes two-dimensional images. There's three major techniques that we use within the scanning electron microscope to create volume data. And let me take you through these methods now. Array tomography is if we took the sample and we cut each slice. So instead of just having one slice, we collect multiple slices, one after another, and we put them all in a ribbon. And we take images of those one at a time. You take those images together and you'll end up with a stack of images. Now, this can be a little bit of front-loaded work as you have to collect all the slices carefully and you have to image each of those slices. Now, on the other side, you can take them into a focused ion beam scanning electron microscope. So that's over on the right there. So the focused ion beam is like a little laser where you can mill a little bit of tissue, and this will actually give you some of the best resolution within that depth. However, the field of view is going to be limited by that technique. And so kind of marrying these two together, right in the middle, we have serial block face imaging. So we could take the block of tissue, mount it in the microscope, and cut it thin with a diamond knife. So this is the same kind of technique you would use in array tomography. If you're familiar with an ultramicrotome, it's like a very small deli slicer. That's how you would collect those slices. We've taken that technology and brought it into the scanning electron microscope. So that diamond knife is able to cut the block of tissue till you have a smooth top surface, and we image that top surface. Then we cut it again, and we image it again. So that block and the top of it is the block face, and each time we take a cut, we take another image. So as you can see in step 2 there, we get one image after another, and if you combine them all together, you get a 3-dimensional volume. So we're doing a serial block face imaging technique. So I could explain this with words, but I think the best is if we just take a look at this quick 2-minute video that we produced to show you the ZEISS Volutome in its entirety. And after we watch that, what we're going to do together live is I've filmed myself imaging and preparing a sample with the Volutome. So you'll get a behind-the-scenes look at how do we collect this data. So let's go ahead and take a look at the Volutome together.
ZEISS Volutome is your in-chamber ultramicrotome for a serial block phase imaging of biological ultrastructure. With Volutome, you can easily transform your conventional 𝗶হি𝗰𝗶𝗹𝘀 to a serial block phase imaging system.
I just want to pause right here and mention this is a really cool part of it. The Volutome is not just one dedicated tool, it's a stage that we bring into the scanning electron microscope. And we could actually swap that out and put in the normal stage, so you have a scanning electron microscope when you need it, and then you also have the Volutome when you need it. By the way, there's a chat over on the right, so if you have any questions during this, I'm here live, and we'll be able to answer those questions together. All right, I'll let this continue.
Let Volutome cut serial sections as thin as 25 nanometers with high precision. Get high contrast images of your delicate biological samples with the sensitive volume BSD detector. Resin-embedded samples are prone to charging. Focal charge compensation ensures the highest image quality by eliminating these unwanted effects. Use mosaic imaging to capture the ultrastructure of your sample in a larger context. Let Volutome acquire thousands of images over hours and days, automatically and unattended. Enjoy integrated processing and visualization of your acquired data, and reveal the details that answer your scientific questions. ZEISS Volutome is your end-to-end solution that integrates all hardware and software components for serial block phase imaging. Want to learn more? Visit our website.
Wow. So hopefully now you have a good idea of all the components and all the engineering and hard work that went into the Volutome. Now, let's take a step back and remember that we're talking about the upcoming Society for Neuroscience conference and how is this going to help neuroscientists. So here's a great example. We have, courtesy of Professor 𝗠cie𝗿𝗃 at the University of Lausanne. She has a Volutome in her lab there. And the data spinning right behind me is some data that they collected in their lab. So this is mouse brain tissue that they brought into the Volutome. And this is the perfect technique for this kind of data. Now they have a large volume where they want to acquire the data through this volume that you see here, and they want to understand what the neuronal network and structure of the brain is. And they also want to capture the unpredictable paths of these cells. And this is a big challenge in neuroscience, is connecting all the different parts of the brain and how are they wired together. Now, with this technique, we get details not just on single little lines of where those axons and neurons and all those synapses are, but we actually get subcellular details. So we could start asking questions of what are the mitochondria doing inside these neurons and how many synapses are coming off of these branched, very complicated neuronal processes. And so the Volutome is going to be giving us this large volume through this tissue here. Now, as I promised, I want to give you a little bit of behind the scenes look of what does it take to start getting these data. So this was yesterday. I've put on a sample to prepare for our demonstrations we're going to do at the Society for Neuroscience conference virtually. And I'm grabbing the sample pin. So that's in the back there. You can see I could keep a variety of different samples on the pins. And this gives you an idea of the size of the sample we're working with. So we're typically looking at about a cubic millimeter of tissue that is mounted and stained and brought into a plastic resin. So that has been prepared ahead of time, and I put it on top of this pin, and the pin simply slides into the sample holder. And now I'm tightening the set screw to lock that pin in place. Now, before I could bring this to the Volutome, I have one more task to do. I need to center this pin to make sure that it is facing the right way and it will be set up well for when I bring it into the Volutome. So part of the Volutome solution is this stereo microscope, which has a special stage that you could see that I've put the sample holder into. And what you can't see here, but that I can see through my eyes in the oculars there, is I have a small crosshair. And that crosshair is telling me where is the center of my sample holder. And what I'm doing there is just using my hands to adjust, to spin around the sample holder to get the pin perfectly aligned. Once that's done, I'll take the sample holder out and I'll tighten it and I'll bring it over to the Volutome where I will vent the stage. So here we are in front of the Volutome. The chamber is vented and we pull out the Volutome stage. And now before I'll load the sample, we actually have another stereo microscope. Now this one is special because it's designed to be mounted on the door of the stage. So we have a specific area where that tightens in, and then I'll go ahead and grab the stereo microscope. And I want to point out that this has a camera on top as well. So this is going to let us, through the software, get a view of what's going on. Because remember, the sample pin is coming to a point. Our sample is about 1 millimeter wide, and I'm going to need to approach the diamond knife that is inside of the chamber. So this really helps me do my job. Of course, we have some gooseneck lights that are integrated into the front of the door there, and I could turn that on, giving me the illumination I'll need. Now I'll move the camera closer, and we could get our first view of the actual Volutome stage. Now, as I zoom in, you'll notice closest to you, here, you'll see the knife on the left-hand side. And the sample pin isn't in yet. That knife is in its parked position. And now I can bring in the sample just like this. So the sample holder has only one way it fits in, and this is making sure that the alignment I did is good. And I just have a simple hand-tightened screw that I am applying here to secure the sample. And finally, I'll place the focal charge compensation needle pointed directly at the sample. Now here's something really cool. See what I have in my hand there? That is there it is. That's a Xbox controller that we've specially designed with the Volutome logo on it, of course. And I can use this for the approach of the sample to the diamond knife. So if any of you in the audience are familiar with working with an ultramicrotome, this is exactly like that final step of approaching the sample to the diamond knife before you would collect the sections. However, this time I'm just doing it within an SEM stage. Now, what the controller is allowing me to do, so I'll play the video here, take a look at the screen on the right. As I press back and forth, I actually can move the position of the knife over the sample. This is allowing me to get perfect approach of the sample to the knife, and also defining that cutting window of where do I want it to start carefully cutting and where do I want it to end cutting. I have, with another control on the controller, the ability to adjust the height. So remember, as we're going to take these cuts, it's not the knife that cuts into the sample by moving down. The knife is always going to stay on the same level, and the sample moves up into the knife. And we'll get a really nice view of this right here. So this is through that camera that's on the stereo microscope mounted on the door, and we can actually monitor the cutting. So here's the first cut I've taken off the top of that block face, and we see it collect on the back side of the diamond knife. Now, each of these cuts are only 50 nanometers thin. That's over 2,000 times thinner than one human hair. And that is what's going to give us that great resolution in the Z dimension. Here's one final cut, just polishing off that block face. Now, that really precise motor in there is what allows us to get that Z depth, and we can actually cut as thin as 25 nanometers once we have the sample in. Okay, I've now closed the door of the chamber. I've taken that stereoscope off, and I've pumped it down to high vacuum. So in an electron microscope, it's all in high vacuum on the inside, and we have a camera inside the chamber allowing us to take a look at what's going on. Now I'm pointing out, there's the FCC needle, there is the sample, and on the right you see the knife in its parked position. Now if I go over, turn the beam on, I actually have a live view of the block face inside the SEM chamber. We see the focal charge compensation needle on the left, and I could just simply double click anywhere in this live view to center the block face. Now, the detector I'm looking at here is just simply my secondary electron detector. That's just showing me rough surfaces, but it's not going to give me the detail I need. For that, I'm going to have to insert my volume BSD, or my volume backscatter detector. So this is a specially designed backscatter detector that ZEISS has engineered for low kV imaging, as low as 1.2 kV. So I've given the command to insert the detector, and we'll see it come in from the right-hand side of this live view. Boom, there it is. Now let me switch back to imaging with this detector. So there's the secondary electron view, and now here is the images from that volume BSD. We now can see the neurons and the cells and all the details inside. Here I'm just doing my adjustments of focus and stigmation and brightness and contrast, zooming in a bit to make sure everything's looking good. I can navigate around by just double clicking on the sample. I'm going to zoom in here and do one final check of my focus and my stigmation and zoom out. I think this looks good, and I'm going to be ready to start collecting some data. So there we see, if I slow the scan speed down, I get a really nice clear picture of the cells and the detail I need. And so this is in that final setup steps for collecting data. And now this is what it looks like to actually collect data. Now this video clip here has been sped up about 3x. If you look in the top left, it's pointing out that there's time it's spending to cut. The knife moves out of the way, just like you saw in that first video, and then it images. And then we see another image. And it will come and take a cut and take another image. And so this is data acquisition in progress. Another step that you don't even see behind the scenes is it can register these slices to each other. So this is an end-to-end solution that when you export the data, not only do you have all your slices, it can export it as one single file that has all the slices, and those slices have been registered to each other as well. And this is just the simple stack. We can, of course, do larger mosaics and even sub-areas within the area of the block face to get multiple fields of view. And we can end up with data that looks like this. Now, this is courtesy of Nickmer at UC San Diego. This is a serial block face data set of brain tissue, and there's just a mind-boggling amount of detail going on here. We see different cell bodies, we see different axonal processes. Every so often, we'll even see a synapse as those two processes come together. We see some postsynaptic vesicles there. There's just so much going on here for a neuroscientist to dive into and learn about, and this is why this technique is just so great, that you have so much detail across such a large area. Let's take another look at the data that we saw earlier. Here's just a deeper dive into this tissue and all the different kind of information that you can pull out from this volume. We can segment the blood vessels, right? And this is all done in ZEISS's 𝗶𝗳 software, where you can take the data you get from the Volutome and you could really start asking a lot of questions. So there's the myelin coating around those synapses. We can look at a single cellular body and the size of its nucleus and what's going on in the nucleus. We could look at it from the other dimension because this is fully a three-dimensional data set. We could then take away the data and just look at what's left with the segmentation. And you can pull out all these different kinds of views, measurements, and if you repeat these across different areas, you can really start to think about the experiments that are possible with this quite amazing tool. There's a node where you have one myelin sheath end and the next one begins. This is a very fine structure within the cell, and malfunctions of those are related to several diseases of the brain. We could even fly inside a single axon and fly out of it. So kind of to summarize here, for the Volutome, I want to point out just that why this is such a great tool is that it has the ability to acquire these large 3D data sets. And let me move myself out of the way here. That we can get these big data sets. This is an ultramicrotome mounted inside your SEM. And the tiles that you can use are up to 32K pixels in resolution, and then we can even mosaic that. We have superb image quality thanks to that volume BSD detector, and of course the focal charge compensation that is putting nitrogen just where we need it, allowing us to look at a whole variety of different samples within this plastic resin that normally is really hard to image within an SEM. And it's fast, and you don't have to sit there and collect each slice. Once you press go, it is collecting those slices in a fully automated fashion, and then, as I mentioned, it's processing that data as well. And of course, this isn't a dedicated tool. This is just something that can sit on your SEM when needed, and then you could take it off and you could put on a regular SEM stage for all the other kinds of SEM operation modes that you need. So I do want to remind you before I go to come see us at the Society for Neuroscience Conference coming up this Saturday. I'm very excited for it and excited to be showing this tool off there. If you have any questions, you could put them in chat. Otherwise, please check out some upcoming workshops that we're doing. We have training videos, especially if you're one of our customers already and you want to learn more or get some of our latest software. That's all going to be through the ZEISS portal. And of course, some of our upcoming streams every Thursday here on our YouTube channel. So with that, I'll wish you a good day and a good Society for Neuroscience conference. I'll see you later.